my pleasure to introduce today's seminar speaker, Kevin Coleman-Dare. He is a computational biologist at the Joint Genome Institute over in Walnut Creek. And he got his PhD from UC Berkeley, 2012, working with Daniel Zilberman. And today he's going to talk about his work on agave microbiome, and specifically an interest in application of agave for biofuels. Kevin?
Thanks, Trevor. It's a great pleasure to be here today to speak with you about this work that we've been doing. As many of you probably know, the EBI has been funding and also conducting research for 5 years or more on investigating whether the agave would make a good biofuels crop. And it's that work and work like it that sort of inspired the research I'm going to tell you about today. So the presentation I'm going to give you can kind of be broken up into 6 different parts. We're going to start off by talking about why we're interested in agave in particular. For the purposes of this audience, this may be less relevant. You guys probably understand that for the biofuels industry, we need to look at a lot of different potential feedstock crops and not just one. So exploring all these different avenues is a very important part of that research. Next, I'll talk about the research questions that we attempted to address with these experiments. I'll tell you a little bit about the sampling plan, so which agave species we looked at, where we collected them, when we collected them. I'll talk to you about the data itself, which is, in this case, 16S and ITS2 microbial community profiling on the MySeq platform. I'll tell you about some of the conclusions I think we can draw from this data. And finally, I'll allude to future directions for this project. So why agave? So if we look at a map of biofuels feedstock crops and where you can grow them in the United States, it's obvious that there's a very large portion of the western half of our country where we don't really have good candidate biofuels crops that can be grown there. And the reason for this, as most of you are probably aware, is that the annual average precipitation across this portion of our country is very low, in some cases less than 5 inches per year. So these are semi-arid and arid lands, and some folks have been wondering whether it might be possible to do biofuels production on these lands. And the reason for that is if we look at the other half of the country, all of these places where we could grow switchgrass, miscanthus, poplar, etc. These are, for the most part, arable lands where we're already producing food staples, such as soy or corn or wheat. So by growing biofuels crops out here, where nothing else likes to grow, we would prevent displacing the crops that are already being used for our food supply. So one possible solution to this, given that I've told you that none of these other crops really like to grow here, would be to look at crops that are native to these regions. So one such crop is the agave. There are roughly 200 species of agave that are native to the southwest and parts of Mexico. They come in a wide variety of shapes and sizes. The smallest is just a couple of inches tall. The largest can be up to 4 meters. And that's not including its flowering stalk, which reaches an impressive 12 meters in height. So there's a lot of different species, most of which have not been extensively studied. So part of the work in understanding whether agave would make a good biofuels crop will be investigating these lesser-known cultivars. So most of you probably know that the agave has been cultivated for hundreds of years, if not thousands of years, in the Americas. There are actually 2 industries associated with agave. So once you've harvested your agave plant from these plantations, you sort of split it up into 2 different parts. So you have the large sort of orb-like stem section known as a piña, and you have the leaves. And what you do next depends on which industry you're working in. So the leaves are harvested for fiber, also known as sisal. And over the last 100 years, this industry in some cases actually outpaced the tequila industry. So this was a big deal for a long time. The other industry, which I've alluded to, is to take the piña, to bake it, ferment it, and turn it into various kinds of alcohol, such as tequila or mezcal, etc. Unfortunately, in the last 20 to 30 years, the advent of synthetic fibers has really displaced the sisal industry. This been estimated that approximately 0.6 million hectares of unused agave plantation are just sitting there, sort of fallow, waiting for something to be done. And this is a problem not only for this infrastructure, but also for the labor force. And this affects parts of Mexico, but also other parts of the world, including Africa and South America. So some folks have been wondering whether we might be able to turn this unused agave plantation into a biofuels industry. We already know how to get alcohol out of agave. It's been shown that you can do something similar to make a cellulosic ethanol. I'm not really going to talk much about all the work that's being done to determine whether it's economically feasible to extract ethanol from the agave. There are plenty of groups working on that. Another group, actually some of the people who are part of this project, did some other research where they created spatially explicit site suitability models for the agave plant. They took predicted temperature ranges across the United States. They took soil chemistry measurements. They looked at irrigation supply. They looked at transportation. And they built these into one model that predicts where we can grow specific species of agave and have it be an economically feasible biofuels feedstock. So the story I'm going to tell you about today is related but quite a bit different from these. So we know that the agave is well suited for arid environments. There are a number of different plant adaptations that make it a successful desert plant. The first of these is a special form of photosynthesis known as crassulacean acid metabolism, or CAM. The trick here is that the plant is partitioning its uptake of CO2 and fixation of CO2 and its actual photosynthetic reactions into two different parts of the day. So at night, when temperatures are cooler, it pulls in the CO2. Then during the day, when things get really hot, it closes its stomata, thereby losing less water back to the atmosphere. Another really cool feature of the agave plant is hydraulic isolation of roots, also known as contractile roots. So during periods of drought stress, the agave roots shrink away from the surrounding soil particles, and it's the points of contact between the soil particle and the agave root that draw moisture out of the plant. So this helps, again, prevent moisture loss. An additional adaptation that the agave has is a very thick, waxy cuticle. So the waxy cuticle is one of the outer layers of the plant leaf surface. In the case of the agave, this can be up to 25 times thicker than the average cuticle in other plants. And actually, in some parts of Mexico, they peel this off and use it as a food wrapper for certain traditional Mexican dishes. So we at the JGI have been interested in whether there might also be a microbial component to the agave's success in the desert. So it's been shown for many decades now that there are plenty of microbes, both bacteria and fungi, living on the surface of the root and in the root that can contribute positive phenotypic effects to plants growing in periods of stress, environmental stresses of all kinds, biotic or abiotic. Some of the ways these microbes can help the plant or promote plant growth are, number one, they can compete with pathogens, either by producing antimicrobial compounds or inducing the host defenses through systemic acquired resistance, thereby priming them to be ready when a pathogen attacks. Another thing that they do is some of these produce phytohormones, such as auxin, gibberellin, or cytokinins. They do this either by encoding the gene products that make the hormones themselves or inducing the plant host to do this. Additionally, they help with nutrient exchange. So nitrogen fixation is one that probably most people are familiar with. You have the rhizobium and the arbuscular mycorrhizal fungi who fix nitrogen for plants. They also do things like solubilize phosphate in the soil. Some of these secrete siderophores, which help to chelate iron from the soil. They can also improve water uptake. And then lastly here, it's been shown that some of these beneficial microbes can alleviate abiotic stress response. And the mechanisms behind this are still trying to be unraveled, but it's known that you can add certain plant growth promoting bacteria to a plant under drought stress and see improved vigor in the presence of that microbe. So all of this sort of led us to form the Agave Microbiome Community Sequencing Project, or CSP. There are 4 different teams working on this. I'm going to go through the individual contributions of some of these team members during the acknowledgments, but I thought I'd point out that there's the team at JGI in Walnut Creek who's doing the sequencing and also analysis of this data set. There's Gretchen North at Occidental College in Southern California, who's collecting some of our agave species. There's Dr. Leila Partida in Sinvestive in central Mexico, who's collecting some of the rest of our agave species. And lastly, Kristen DeAngelis, who's a rhizosphere expert we brought on board to help us interpret some of these findings. So the project actually consists of two phases. Phase 1 is what I'm going to talk about today. So this is a microbial community profiling, both for bacteria and for fungi, across several different species of agave and across several different locations and 2 different time points. Phase 2, which I'll talk just briefly about at the end, consists of isolate sequencing and also functional testing for some of the candidate symbionts that we have identified. So in terms of research questions, when I first put this slide together, there were like 15. It was really messy. And I decided to make it easier both for me and for you and try to umbrella these under 2 larger, broader questions. So question number 1 is, what are the principal drivers of microbial community composition? And question number 2 is, what microbial taxa are enriched in the agave microbiome? That's with respect to the surrounding soils or with respect to other plants growing in the desert. I'm going to tell you a little bit about our sampling plan. We actually collected from 6 different sample types. The leaf episphere, also known as the phyllosphere, is those microbes growing on the surface of the agave leaf. We looked at the leaf endosphere, microbes inside the agave leaf, the root endosphere, and the rhizosphere. So the rhizosphere is the surface of the agave root and the 2 millimeters of soil that adhere to that root. In addition, we used 2 different soil controls, one which we called root zone, was taken 10 centimeters away from the agave root, and bulk soil was 1 meter away from the agave plant. For the purposes of today's talk, we can kind of think of grouping these 6 different sample types in a couple of different ways. So one might be above versus below ground. So we have the aerial tissues, we have the root tissues, and then the soils. Another would be the episphere versus the endosphere, so the surface of the plant versus the internal compartments. We looked at 3 different species of agave. So Agave deserti is native to Southern California. It's one of the smallest species. It's only about this long. It is also the most thermotolerant of all known agaves. It can survive temperature drops to -4 degrees and up to 130 degrees Fahrenheit. Agave salmiana is the largest of the agaves. It grows to be somewhere around 4 meters in height. It's found in central Mexico. And Agave tequiliana is the one that you're probably most familiar with. So this is the one that's classically cultivated in the production of tequila. It doesn't exist in the wild. This is a cultivar that's been bred for many generations, and you really only find it in the context of plantations. Additionally, work that I'm not really going to focus on too much today is centered around 2 other non-agave species that we collected, Myrtillocactus geometrizans and Opuntia robusta. They grow in the same region as Agave salmiana. We use these to try to establish which microbes are enriched in the agave microbiome with respect to other desert forms. So in terms of collection sites, we actually had a total of 9 different sites that we looked at. So Agave deserti, which is native to Southern California, we collected from the Boyd Deep Canyon Preserve. There were 3 different locations, Pinyon Flat, Agave Hill, and Boyd Ridge. These are separated by about 1 kilometer, and there were changes in elevation associated with these sites. Agave salmiana we collected from central Mexico near Guanajuato. 2 sites, Penjimo and San Felipe, were sort of natural parks. Agave tequiliana we collected from 2 different tequila agave plantations, El Magueyal and Amatitan. And the cacti that I described to you we harvested from Las Animas and El Jaral, which were located very close to where we found the Agave salmiana. I'm going to spend some time today talking about differences that we see in microbial communities between the cultivated and wild species. So I thought I would point out here, we have 1 cultivated species, Agave tequiliana. Everything else was taken from its natural habitat. Additionally, we sampled twice. So we sampled once in spring and once in summer of 2012. So there were a lot of slides there, and I want to just give you an overview of this sampling plan. We can think of this in terms of 3 different axes. So axis 1 is sampling type. You can sort of partition these sampling types in different ways: above ground, below ground, inside, outside, depending on what you're interested in looking at. We can also think of the sampling site as another axis. So here we have California versus Mexico, 2 different regions. We have 3 different host species: Agave deserti, Salmiana, and Tecolana. We have cultivation status, wild versus cultivated. And we have the subregions, which I mentioned on the previous slide that had a map. Additionally, we have another axis, which is sampling time. So if we put all these together, we ended up with 6 sample types, 9 subregions, 2 seasons, and then in each of those permutations, we did 3 plant replicates. So that's a total of 300 different samples that we looked at. So once we'd collected these samples from the field, brought them back to the lab, extracted the DNA, we did 2 different microbial community profiles. So the first, to understand the prokaryotic communities, used the 16S ribosomal subunit, specifically the V4 region of that gene, which these primers target all of the bacteria in archaea. For fungi, we used the ITS2 region, which stands for internal transcribed spacer. This is specific just to fungi, doesn't amplify from the rest of eukaryotes. I wanted to walk you through just really quickly in one slide how you make a next-generation sequence tag-based microbial community library. So you start off with your samples, soils, whatever. In our case, we have 300 of these. Each of these has its own sort of microbial species living in it. You extract the DNA, and then using the primers that I described, you amplify out your region of interest from your phylogenetic marker gene. So in our case, 16S or ITS2. Then through sequencing, clustering, and taxonomy assignment, you can build what we call an OTU table. So OTU stands for Operational Taxonomic Unit. These are sequences that clustered together at 97% identity. And you can kind of think of this as a species that we saw in our given environment. So the data matrix here is what's used for all of the downstream analyses that I'm going to describe to you. So it's very simple. Each of the columns represents one of our samples. Each of the rows represents an OTU or species that we saw in some of our samples. And then the cells in this data matrix represent how many times we saw OTU X in sample A. So there are a number of advantages associated with this kind of approach over the clone-by-clone culture-based approach of the past. So number 1, it's very deep sequencing. We can get 100,000 different species or OTUs in one sequencing run. It's very fast, so you can do all of this in one afternoon on a MySeq. It's inexpensive in terms of a read per read basis. So I'm going to give you one slide that sort of summarized all of the read statistics for the reads that we generated. In total, there were 72 million MySeq reads. These are 2 by 250. So we have a forward and a reverse from our region of interest. These end up being merged together. So you end up with one amplicon for your 16S or your ITS2 region. This equates to around 100,000 reads per sample. And then after doing all of your QC, where you remove bad quality reads, you remove chimeras, you get rid of reads that didn't have a mate pair, you end up with around 25,000 to 50,000 reads per sample, which is still more than enough for doing most kinds of analyses. So unfortunately, these are not evenly distributed across our sample types. So I'm showing you here box plots for read counts across the various sample types. On the left, we have the 16S or prokaryote data set. On the right, we have the ITS2 or fungal data set. And what I hope you can see is that in both cases, the leaf endosphere had a very low average or median read count, on the order of 1,000 to 5,000 reads per sample. So this is a problem for us, because if we want to compare across these, we're going to introduce potentially some statistical bias associated with differences in read depth. So we had to do some pre-processing and normalization to account for this. One thing that we did is, number 1, we got rid of OTUs which were very low abundance. So if a particular OTU only had one read and we only saw it in one sample, we we kind of counted that as noise and threw it out. So we only wanted to keep the measurable or high abundant OTUs. Additionally, we had to do some normalization for read depth. So we did this by randomly subsampling 1,000 reads from each sample. By doing that, we're able to then still do a comparison with leaf endosphere, root endosphere, etc. And this means, of course, that these extra 25 to 50,000 reads, we're losing a lot of our sequencing power there, but what we've actually found is that it doesn't really change the overall picture. So I've done some analyses where I've increased that number, and we don't really see major differences in terms of the overall story. Okay, so now the actual analyses that we've done. Question 1 that I mentioned before is, what are the principal drivers of microbial community composition across these samples? Specifically, does it matter if we're looking above or below the ground? Does it matter if we're looking on the surface of the plant versus inside? Does host species play a role? Does cultivation status? And lastly, but certainly not least, are there different drivers when you're looking at the fungal communities and the prokaryotic communities? So there's a lot of different ways that one could ask this question or answer it. I'm going to show you here some principal coordinate plots. So basically, this is all 300 of our samples, and I've colored them here by either soil, episphere, or endosphere. We're looking at the prokaryotic data set, and what I hope you can see is that there's a nice clustering of soil on the left, endosphere on the right, and the episphere in the middle. So this is sort of intuitively what you would expect. You have the outside, you have the surface, and you have the inside of the plant. If we look at the fungal data set, it's kind of a different picture. So again, this is not random clustering. There's definitely groups that appear to be clustering by this particular factor, but it's not as clean as what we're seeing for the 16S data set. So for principal coordinate analysis, obviously, there are many different axes that one can plot. I'm only showing you 2 of them here. So one criticism of this might be that, well, what happens if you plot other axes? Can you see some better separation by this factor? Rather than plotting out all the axes, we can do a different statistical test. So this is analysis of similarity, or ANOSIM. So ANOSIM gives you one statistical value that describes the degree of clustering you have across a particular factor. So the values, you can think of these ranging from 0 to 1. 0 would be complete randomness, no clustering at all. 1 would be perfect clustering. The ANOSIM statistics for these two are 0.5 and 0.237. Anything above 0.1 is considered meaningful degree of clustering. So both of these show non-random clustering, although you'll notice that the value for the prokaryotic data set is higher, which suggests that prokaryotic microbial communities are more influenced by inside versus outside the plant than the fungal data set. We can do the same sort of analysis using the above versus below ground factor that I described earlier. Again, for the bacterial data set, we see a nice separation of the aerial and the root. Over here, there's a good deal of separation, but there's also this sort of area here where you have a lot of overlap. And the ANOSIM statistics, again, reflect this, where the prokaryotic data set has a higher ANOSIM stat than the fungal data set. So far, we're only looking at sampling type. If we look at sampling site, do we see something similar? So I'm showing you here by region, which is California versus Mexico. And if we look first at the right, you can see that you can actually draw a vertical line through this plot that perfectly bifurcates those samples taken from California and those samples taken from Mexico. On the left, for the prokaryotic data set, that difference is not as clear. There's a lot more overlap. And the ANOSIM statistics reflect that. We could also do this by host species. So here, again, Agave deserti from California stands out on its own. Agave tequiliana, we start to see separation from Agave salmiana. In the case of the prokaryotic data set, that separation is not as clear. So that's a lot of plots to look at and to keep track of. So another way we could do this would be to just plot the ANOSIM statistics themselves. So that's what I'm showing you here. These are the ANOSIM statistics from the previous slides for the fungal data in red and the prokaryotic data in blue. The first 3 sets of bars are all factors associated with sampling type, right? And in each of these cases, we see that the bacterial microbial communities are more influenced by sampling type related factors. The next three factors, or sets of bars, are all related to sampling site, so host species, subregion, or region, and in each of these cases, the anosim statistic was higher for the fungi than it was for the prokaryotes. One other thing that I'll point out, which I thought was kind of interesting, is that in terms of overall microbial community composition, season appears to be playing almost no role at all. In fact, it's closer to complete randomness than what we got for replicate, which was unexpected. So conclusions that we can take away just from this first part are that, number one, fungal communities appear to be more influenced by sampling location or host species, and bacterial communities appear to be more influenced by sampling type. Additionally, season seems to play an almost negligible role in determining overall community structure. So the second question that we were interested in addressing, which is sort of the broader question, is which taxa are enriched in the agave microbiome? So by this, I mean enriched with respect to the surrounding soil or enriched with respect to non-agave species. In particular, we were interested if there's a selection process happening here where the agave plant is pulling out microbes from the soils that it wants to enrich in its environment. And is this selection process similar between different agave species? Additionally, does cultivation seem to affect the microbial community? So to start with, I'm going to show you a couple of Venn diagrams. So here, the numbers in here represent all of the genera that we detected for prokaryotes on the left, fungi on the right, for the three different categories of sample types: soil, episphere, and endosphere. And just as a quick reminder, episphere, those microbes growing on the surface of the plant; endosphere, microbes growing inside the plant. The values that I want you to pay attention to are these. So if we look at the number of genera shared between soil and episphere, we see that that's actually quite high, 124 and 106. As we look at the number of genera shared between episphere and endosphere, that number decreases. And when we look at the number of genera shared between endosphere and the soil that weren't part of the episphere, that number is very close to 0. So this is consistent with a model whereby those microbes living in the endosphere are a subset of what you find in the episphere, which is a subset of what you find in the soil. So you can think of this as a gating effect, where the plant is, at each stage, as you move closer into the plant, it's selecting certain microbes out of that environment and allowing them to pass on to the next level. Another thing we can do is we can just pull out all of the genera that we find in the soil. Right? You'll see that there's still a large number of both prokaryotic and fungal genera that we find in the plant that we're not detecting in the soil. So does this mean that these are things that aren't present in the soil at all? I don't think that that's likely to be the case. Remember that we've normalized to a constant read depth of 1,000 reads. So what it means is that these particular orders are enriched in the plant and at our detection level are not detectable within the soil. So they represent less than 0.1% of the microbial populations there. So another way to sort of get at who those microbes are, the ones that the plant is selecting for, would be to look at some relative abundance plots. So here we're showing order level relative abundance for prokaryotes on the left, fungi on the right. I'm not going to go through all of the different taxa here. It's probably too difficult to explain all of those things, but I want to point out two patterns. So number one, if we look at the fungal relative abundance plots, we see that there are particular fungal orders that are increasing in relative abundance as you move into the plant. So from bulk soil into the root zone, rhizosphere, root endosphere. And this increase in relative abundance is happening at the expense of other fungal orders which are decreasing as you move into the plant. The same thing is seen in the prokaryotic data set. So bulk soil and root zone, the profiles actually look very similar to one another. But as soon as you approach the rhizosphere or move into the plant, certain bacterial orders are taking over those environments. And I want to focus on those two in particular here as sort of an example. So these two orders are Pseudomonadales and Enterobacteriales. One question that I had when I saw this is this distribution sort of similar across all rhizosphere samples? Or does it change from rhizosphere sample to rhizosphere sample? So one way we might start looking at that is to look at reed counts from the plant-associated samples and the soil-associated samples for these two different orders. And what we see in both cases is two things. So number one, the median or average number of reed counts for plant-associated samples is much higher in both cases. But number two, there's a large sort of degree of variation in the reed counts that we're seeing here. It's not as if all of them have exactly 20% of their relative abundance attributed to Enterobacteriales. There seems to be quite a bit of variation. So I started to wonder whether this variation might be associated with the host species or cultivation status. So one way to look at that would be to just focus on this bar, the rhizosphere bar, and instead of doing an average profile, do a profile for all of the different rhizosphere samples we have and line them up next to each other. So that's what I'm showing you here. This is every rhizosphere sample that we collected. I've ordered them by the host species, Agave deserti, Salmiana, and Teccolana. And what I hope you can see is that the cultivated agave actually has a very large percentage of its relative abundance as part of the Pseudomonadales and Enterobacteriales families. So someone in an audience for this talk that I gave recently asked, well, maybe this is just something that's happening in the soils around these plants. It doesn't necessarily have to be something that's specific to the rhizosphere. So we looked at the bulk soil profiles for these samples. And we see that the bulk soil sample profile looks actually fairly stable, right? So this effect that we're seeing here, where certain bacterial orders are swamping the rhizosphere, appears to be a rhizosphere-specific effect. So this discovery kind of led me to investigate whether other plant-associated sample types showed a similar distribution. So if we look at the leaf episphere, we see something similar. So in the case of the cultivated species, 2 or 3 different bacterial orders have sort of taken over the episphere, in particular Flavobacteriales, Pseudomonadales, and Enterobacteriales again. And what this indicates to us is that the natural microbial diversity, at least in terms of the bacteria, associated with the epispheres of agave is being lost during the cultivation process. Another way to assess microbial diversity is through an alpha diversity metric such as Shannon's, which takes into account both the number of species you see in an environment and the evenness of their distribution across those samples. So here are the Shannon's entropy values for the rhizosphere on the top and the leaf episphere on the bottom. I've colored them here by the host species, and in all cases you see that the Agave Teccolana Shannon's diversity is very close to 0. So this was a very interesting discovery for us, and we'll come back to it a little bit at the end. I wanted to take just a few minutes to talk about some of the other things that we've done. So we found, for one, a number of different prokaryotic and fungal genera that appear to be enriched within the plant compared with the surrounding soil. So some of these you'd expect, Pseudomonas, Agrobacterium. Some of them were less expected. Lechlercia is a member of Enterobacteriales, and I'll talk more about that one in a little bit. For the fungi, there were also some interesting ones, Penicillium and Oreobasidium, which contain plant growth promoting species. Additionally, we found a number of different bacterial genera which distinguished the agave rhizosphere from the rhizospheres of the cacti. So I'm not going to go through all of these, but I'll point out 2. So Streptomyces and Agrobacterium, both of these are interesting because they contain known plant growth promoting species. Additionally, they also contain known plant pathogens. And with the sequence data that we currently have, we're not able to get species-level taxonomic classification. So we don't actually know if this is Agrobacterium tumeficans, which is a pathogen of many plants, or if it's one of the other Agrobacterium that promotes plant growth. So we'll have to do further work to sort of go in and figure out which of these species is present here. And I'll talk about ways that we are actually trying to do that now. One other thing that we did was to look for known plant growth promoting bacteria. Some of the ones that you might be familiar with are bradyrhizobium and rhizobium, which fix nitrogen. We found that for the agave species, there was an enrichment in the root endosphere and rhizosphere for both of these, along with Burkholderia, which is another bacterial genera that contains many plant growth promoting species. In all of these cases, we see more of them as we move into the plant than to the surrounding soil. So these are the kinds of targets that we would like to identify and isolate from the plant for possible functional trials and work in the field. One last example is Penicillium. Again, Penicillium is a very large fungal genera. It contains a lot of known plant growth promoting species, but also some pathogens. We found that this particular set of Penicillium was not only enriched within the agave compared to the other cacti that we looked at, but it's specifically enriched in the root endosphere. So this is one of the ones that we're really looking for in our isolation experiments. So some conclusions that we can draw from this, what I've shared with you so far. Fungal communities appear to be more influenced by sampling location or host species, and bacterial communities appear to be more influenced by sampling type. Bacterial diversity is greater for wild species of agave than it is for cultivated agave. We're not sure why this is, but this is something that we're really interested in pursuing further. We can also detect both bacterial and fungal genera that differentiate the agave microbiome from the surrounding soils and from other cacti that grow in the desert. And additionally, many of these that we found contain potentially plant growth promoting species that we would like to isolate. So future directions for this project, we are in the process of collecting isolates from the rhizosphere and the root endosphere of Agave salmiana and Agave teculana. Our collaborators in Mexico have pulled out so far all of these. We're really interested in some of these in particular. So I talked a lot about Enterobacteriales, which is that bacterial order that differentiates the cultivated agaves from the wild agaves. We have 8 different genera from this particular order. Additionally, Xanthomonidales appears to be specific to Agave deserti, or there's a lot of them present there. We have 1 of those. Bacillus, which is enriched in Agave salmiana. We have 5 of those. One thing that was really interesting to us is this particular one, Pantoia, which contains 1 species that is a putative causative agent in the leading disease of agave and agave tequila plantations, which is soft rot. It was estimated in 2004 that there was a $200 million cost associated with this disease in the agave tequila industry, and roughly 30% of the plants that they surveyed in one given plantation contained this particular or a species from this particular genera. So we want to go in and try to take a look at this more closely and figure out, is this one of the potential pathogens that's leading to major losses in the agave industry. Additionally, there are 62 more bacterial isolates that are waiting to be characterized. We also have 30 fungal isolates. Our collaborators have started doing some functional testing of these. So they take a sterile agave plantlet and they add back in 1 of these microbes, and they look to see if they can detect plant growth promotion under various different types of conditions. I should mention also that we're picking out some of these to do isolate sequencing. So we'll do whole genome sequencing of candidates that we find particularly interesting, and then hopefully use the functional information therein to further this kind of work that we're doing here. So I want to just take a moment to thank the people who worked on this group and made this possible. Stephen Gross was the lead postdoc for this project at the beginning. He wrote the grant that got this project funded. He's since taken in a job with Illumina, and so I'm sort of carrying the torch forward. Julian Tremblay is the bioinformatician who wrote the scripts that did all of the processing of the raw FASTQ files. Axel Wiesel is the lead PI. Susanna Tringe is my PI. Gretchen North, I mentioned, who did all of the collection of Agave deserti along with a sort of horde of undergraduates. Leila Partida in Sinvestiv, Mexico, along with 2 graduate students, did collection for Agave salmiana and Agave teculana. They're also working on a paper where they've used DGGE to do a similar sort of analysis, but with a different experimental focus. And Kristen DeAngelis is a rhizosphere expert who we're bringing in now to sort of help us interpret some of these results. So I've ended a little bit early. Hopefully that makes you happy. I'll take any questions. Thank you.
Yeah, any questions?
Yeah, within a particular plant that's very close and you have a bunch of them, are you pooling that data, or are you looking at individual plants? I think those vertical columns were individual plants.
They are individual plants.
And how much variation do you see between those
between the replicates? Yeah, it depends on which sample type you're looking at. So bulk soil, we see very little variation. But one of the things that we found is that when you look at the endosphere, plant-to-plant variation is kind of through the roof. So it's possible that we're just not getting enough reads. As in the case of the leaf endosphere, we only have approximately 1,000 reads to start with. Maybe if we did a more deep sequencing of those, we'd be able to see some more stability. But there does appear to be a lot of variation for endosphere samples in particular.
Yeah, I was just thinking, you were talking about functional testing and applications. Has this been done before, and how would you envision this being used?
To my knowledge, it hasn't been done with agave, but it's certainly been done in other plant systems. One of the primary challenges is that in most cases this has been done in the context of adding a single organism into your plant, as rhizosphere or whatever. And what we find is that in the lab, that's great. You can see all kinds of wonderful effects. But when you try to introduce this into the field, soil is one of the most diverse microbial environments on the planet. And competition between other species local there usually swamp out your plant growth promoting bacteria or fungi very quickly. So I think in the near future, one of the things that we're going to have to do is look at consortiums of plant growth promoting species together and to try to sort of stabilize these environments prior to putting them into the field. I saw.
So I'm curious about the separation, the nice separation that you see between fungal and bacteria within the plant and within the environment. Is it like agave specific, or we see it also with other plants as well?
Yeah, so we see this with other plants as well. Actually, the very peculiar phenomenon that I described early on, where fungal communities appear to be more driven by host species or sampling site, and bacteria appear to be more driven by sampling type. That's actually just recently been seen in a paper looking at poplar sample type. So they did a similar thing with bacteria and fungi. They looked in North Carolina and in Tennessee at some different sites there, and they found that for some reason the fungal communities showed a lot more variation across locations than the bacteria ones did. And their explanation for that is that it has to do with dispersal mechanisms. So the fungal spores may not be making it as far, so creating these sort of isolation effects between even very close sampling locations. We're still trying to explore that.
And I have a question. Is there much you can do to compare the relative abundance of your fungal species to the bacteria?
Yeah, so
give an impression of which is dominant.
Yeah, that's work that we're trying to do. There's this is actually one of the first next generation sequencing studies where we've done both bacterial and fungal data set together. There are a few, but they're not very many, and they're not usually at this read depth. So we're in the process of trying to develop techniques to look for maybe co-occurrence associations that happen between bacterial species and fungal species, and to try to draw a relationship between the two. And so far, I don't have much data on that, but it's a good question.
More questions? Well, if not, let's thank Devin and enjoy our lunch.